Ivana Nikic-Spiegel from the University of Tuebingen, Germany. She started off her kind of higher learning in molecular biology and physiology at the University of Belgrade, and then received her PhD in neuroimmunology from Ludwig Maximilian University of Munich, and completed her postdoctoral training in chemical biology at EMBL in Heidelberg. At every stage of her career, she's made really impactful, important findings. As example, during her PhD, she discovered a novel form of neuroinflammatory axonal injury in a mouse model for MS, and got a first author Nature Medicine paper, which is quite a good way to get the best postdoc you want. And during her postdoc, she kind of took a left turn, or maybe a right turn, from what she was doing in neuroinflammatory diseases, started developing expanded genetic code and click chemistry-based methods for fluorescent protein labeling in living cells. And remarkably, she's now, as an independent leader, has combined those two areas of research to what she's doing today, focusing on understanding mechanisms regulating neuroinflammatory axonal injury using advanced microscopy and chemical biology techniques. So I find that really fascinating that you took two really different areas of research and combined them successfully. So today, Ivana will tell us about her group's efforts on expanding the genetic code for live cell fluorescent protein labeling in neurobiology.
Yeah, so hello everyone, and thank you for this nice introduction. So I hope you can all hear me and you can see the slides.
Great.
So yeah, I wanted today to give you a bit of basics and also a bit of historical perspective and some practical guidelines when it comes to using expanding genetic code for fluorescent protein labeling and microscopy. And yeah, before I start, just a bit of a reminder. So how can we label proteins inside the cells? So yeah, on the one hand, we can take our proteins of interest and fuse them to fluorescent proteins. And this has many advantages. So making such genetic fusions makes this genetically encodable, which makes it compatible with live cell imaging. This is nowadays relatively easy to do. Also, these fluorescent proteins cover the whole spectrum, so you can do beautiful multicolor studies. And yeah, this works in most of the cases beautifully and shouldn't be replaced with other methods. However, some proteins of interest actually get affected by adding such types of tags to their N or C termini. And also, if we just compare photophysical properties of fluorescent proteins and small organic dyes, these organic dyes can have better photophysical properties. And that's why we also have an interest in how to basically develop tools to attach these small fluorescent dyes to our proteins. And yeah, there are many methods also here available. So if you work with fixed cells, most widely used is immunofluorescence, where you can use primary, secondary antibodies, also nanobodies. If you're more interested into live cell labeling, you have probably heard about tags such as SNAP, CLIP, HALO tag, also sortase-based tags. And all of them, again, have their advantages and disadvantages. But I guess what they have in common is that most of these tags are also large, and some of them are actually larger than fluorescent proteins, which also makes it very likely that they can affect the function of your protein of interest. And that's why, in general, there is this need to develop minimally invasive labeling tags, so to replace something like this with something as small as you can get. And when it comes to proteins, ideally, we can go to the level of proteins building blocks, which are individual amino acids. And ideally, we can add these fluorescent labels at this level. So this is relevant from this functional point of view, but this is also very relevant for us in microscopy field. And this is very simplified scheme, but what I'm trying to show you here that any label, and especially conventional labels such as antibodies, and here is just the primary antibody conjugated to a fluorophore, will always put fluorophores at a certain distance from your target protein. And you can imagine if you have secondary antibody, this gets even longer. And we refer to this as a linkage error. And yeah, if you're working with conventional microscopy, this is not such a big deal because the resolution is not so high. But now with all the super resolution microscopy techniques, this becomes very relevant. And you actually want to put these fluorophores as close as possible to your target protein. And this is possible with unnatural amino acids and click chemistry. And this is basically what I will tell you today about. So, excuse me. So first of all, we are using genetic code expansion to incorporate unnatural amino acids. So they carry these certain chemical groups, which in this case are clickable. And then if we supply the cells with fluorescent dyes, which are, again, conjugated to clickable groups with click chemistry, we can now get fluorescent protein labeling. And with genetic code expansion, we can actually put these amino acids at any position where we want, and that's how we get site-specific labeling. So basically, there are at least two questions that we have to deal here with. So how do we incorporate such clickable unnatural amino acids, and how do we label them? So to incorporate them, we actually need genetic code expansion. So genetic code tells the cell how to translate the information from the messenger RNA into the amino acid sequence in proteins. And yeah, most of you probably know, so I will go quickly through this, that very critical for this process are these transfer RNA, which are adapter molecules that carry actually amino acids to the mRNA template. And they cannot do this job alone, so they also require enzymes, so tRNA synthetases, which will actually charge the tRNAs with their respective amino acids. And then during the protein translation at the ribosome, through the pairing of codons on the mRNA and anticodons at the tRNA, we actually get this correct translation of the information. So we now want to introduce unnatural amino acids, so something that cells normally do not produce. So how to do that? Well, we need to expand the genetic code, and basically this means that we need to supply some additional components for this translational machinery to work. So in addition to the unnatural amino acid, we also need a tRNA, which will be specific for it. We also need to decide which codon do we want to use for the incorporation of the UAA. And we also now need a synthetase, which will be specific for this UAA. And very important is that these tRNA and tRNA pairs are orthogonal to the host cell, meaning that they will not cross-react with any of the endogenous pairs. And that they will be only specific for the UAA. So how do we find these? Well, if we are just searching for orthogonal pairs, usually we borrow them from distant organisms. So in this case, I'm referring really to different domains of life. So it's actually possible to take some pairs from archaea and introduce them in bacteria or eukaryotes where they will be orthogonal. And it's also possible to take some pairs from bacteria and make them orthogonal in eukaryotes. But this is just then one part of the story. This is where we get the tRNA synthetase pairs. So we also need to find the unique codon. And this is a bit more challenging because, yeah, if you look at this table, we have 64 codons, and most of them are already taken. So they are responsible for coding for one of the canonical amino acids. And there are only 3 of them which are unique in the sense that they do not encode any canonical amino acids. These are actually the stop codons. And among these 3 amber codons, so the UAG is actually the most widely used for the UAA incorporation. And that's why we also refer to the method frequently as amber codon suppression technology. And so now we have everything. We have this unique codon, so we can do site-directed mutagenesis. We can put it in our mRNA of interest at almost any position. And that's basically how we get now the genetic encoding of unnatural amino acids at desired positions in our target proteins. So sounds complicated, and yes, it involves many components, but if you want to now do this inside the cells, there is still no reason for panic. So the way we bring this inside the cells is actually to use plasmids, which encode for these components. So first of all, you need a plasmid which encodes for this tRNA synthetase pair. Then you need your protein of interest with this amber codon mutation. And then depending on your model system, you can use transfections, viral vectors, electroporation, basically whatever works in your model system to bring this inside. And this is not much different compared to trying to express fluorescent protein fusion. So you also need to bring these via plasmid. So the only important thing here with amber codon suppression is that you need this orthogonal tRNA and synthetase pairs, which will make incorporation of UAAs possible. And yeah, the method is not so new in terms of genetic code expansion. So it was first now more than 20 years ago developed in bacteria, then very soon in yeast. And actually, very few years later, it was also shown that this can be used for mammalian studies and including primary neurons. However, at that point, the natural amino acids that were incorporated, they were not suitable for, they were not made for fluorescence microscopy studies. So in parallel and in the meantime, there were developments in the field with aim to develop fluorescent unnatural amino acids, and these are just some of the first examples. And there will actually be a webinar in 2 months where I think at least James will talk about some of the fluorescent unnatural amino acids. So I guess we will also be able to hear about the new development in the field. But what is still a problem for us in the microscopy field is that the fluorophores, which we like to use, such as these Alexa fluorophores, are rather large. And actually making them as amino acid derivatives and trying to incorporate these inside the ribosome during protein translation is actually very difficult because they are very large and ribosomes cannot accept them. So that's actually why we are using this two-step process where we are first incorporating these clickable unnatural amino acids, and we can use this with these orthogonal pyrrolysine tRNA-RS pair, and only then with click chemistry we are bringing the fluorophores in. And here I just want to repeat what is very important for live cell labeling is that these click reactions need to be fast, specific, and not toxic for the cell. So if we look at the available options, the most famous alkyne azide click reaction is not the best choice because it requires copper and most of the cells don't like it, so it can be toxic. So that's why we are actually using copper-free reactions which are fully biocompatible. And here we have two options. So one of them relies on using UAAs which have strained alkynes and they can be conjugated with azides. And we have also UAAs with strained alkenes, which can be conjugated with tetrazymes. And both of these reactions are fully biocompatible, so they proceed at physiological conditions, and they just differ a bit in their speed. Actually, not a bit, actually quite a lot. So these tetrazyme ligations are much faster than azide ligations. Also, what makes these reactions very interesting is that they are orthogonal to each other. And this means that they can also be used for dual color labeling. And this was one of the first examples where this was done in bacteria. So you can see here two populations of E. coli which were labeled with this tetrazyme and azide ligation. And actually at this time, this was a very exciting period in this field. A number of papers appeared that actually showed how this type of unnatural amino acids and how this type of chemistry can actually be performed in living mammalian cells. And that is also when I made this change in the field and when I joined Lemke Lab to work on this. And the main focus of my work was trying now to figure out if we can use this click chemistry as a labeling tool for dual color super resolution microscopy in mammalian cells. And at that point, we were working with insulin receptor, and we first wanted to reproduce what I just showed you in bacteria, trying these two ligations, tetrazyme and azide, and trying to get labeling of two populations of insulin receptors. But because of the difference in their kinetics, this was actually half successful. So this tetrazyme ligation worked, but the azide ligation was too slow. And by too slow, I mean that we needed to wait more than 2 hours to get protein labeling, which is rather long if you want to get like fast and live cell labeling. So that's why we then spent some time trying to optimize this, trying to also find two fast orthogonal reactions, both of them based on tetrazymes. And in the end, we managed. So here is just an example. This is the surface of a cell. Here are two populations of insulin receptors labeled with these two chemistries. And we did it in a pulse chase manner, meaning that this is same protein, but labeled at different time points. And at the same time, this was also the first proof showing that this type of labeling can actually be used for super resolution microscopy studies. At the same time, we and others were trying also to establish this for intracellular proteins. And this is a bit more challenging, and I will now try to briefly explain you why. So if we just speak about genetic code expansion, as long as you provide all the components, there is actually no limit in which type of protein you can get these UAAs incorporated in. So this can be a transmembrane protein or cytosolic or nuclear protein. But actually what is limiting are the fluorophores, because if you want to label extracellular something, you need cell impermeable dyes. And most of the dyes are actually cell impermeable. And it becomes very challenging if you want to do live cell labeling, because then you need cell permeable dyes. And this was particularly challenging when it comes to super resolution microscopy studies, because there are really very few fluorophores that actually fulfill these criteria. And in 2013, one of these was developed. So this is the silicon rhodamine dye, which was then also a few years later used for genetic code expansion-based live cell labeling. We were also using this, so I was also using this during my postdoc, but we noticed then something which we at first didn't understand. So independently of the location of our target protein, we actually noticed a nuclear background. And this made us to investigate this further, and we actually could show that this orthogonal fluorescent tRNA pair, for some reason in this conventional system, they get mislocalized in nuclei. So here you can see a nuclear stain, you can see immunofluorescence for the PLRS and then the FISH for the tRNA, and you can see that both of them are in nuclei. And that's not where they should be, because actually protein translation happens in the cytoplasm. So we tried then to get them out and we fused them to the nuclear export signal. And you can see here that we succeeded. So now they are in the cytoplasm where they should be. And this also led now to the increase in the efficiency of the amber codon suppression. And in general, and this is still true for the field, you know, we are constantly trying to improve it to get higher efficiency, better incorporation of the UAAs, and there are several strategies. I will just show you now some of those that appeared at around the same time. So, for example, by using this engineered eukaryotic release factor 1, you can also increase the efficiency, and this factor is important for the termination of the translation, and because we are here dealing with stop codons, is also very relevant. It is also possible to make designer tRNAs, which are then more stable and which also can lead to increased efficiency. And there were also newer studies published last year trying to design tRNAs, which will give us better amber codon suppression with pyrolysing system also. And yes, when I started my group, I wanted to bring these two things together because, yeah, many neuronal proteins, such as ion channels or transmembrane proteins, they're very sensitive to the size of the labeling tag. And I thought, yeah, this is perfect. Let's just combine it. But, you know, I try to show you that already going from prokaryotes to eukaryotes, you know, it's not that straightforward. And now trying to do this in primary neurons came with additional challenges because just biologically, this is a much more complex system. But in 2021, actually, two preprints appeared at around the same time. One of them was from my group, where we actually managed to show that this type of labeling can also be achieved in primary neurons. And I will now just quickly go through it. So what is basically possible, what we did, and what is possible with this method. So in our study, we were focusing on neurofilament light chain. So this is one of the cytoskeletal components of neurons, actually the most abundant. It belongs to the intermediate filament family. These are heteropolymers, and actually these major filaments have a very small diameter. And we are interested in their response to neuronal and axonal injury. And when we tried to label them with GFP and induce this injury, we had some problems with GFP-based labeling. And this is basically what made us to try to make this work with the click chemistry. And this involved optimizations at the level of transfections and at the level of the click labeling. And initial experiments, we actually did fixation before going to the microscope. And this was one of the first examples where actually we got very successful labeling of neurofilaments in living neurons. So we were using silicon rhodamine tetrazyme that I previously mentioned. And after the fixation, we also did co-staining with the flag tag just to make sure that the labeling is specific. So I already told you that one of the advantages of click labeling is that it can be done in living cells. So of course, we then wanted to check if we can also do live cell labeling. Will these filaments be stable? Can we image them over several hours? And yes, this was also possible. So this is just one example of now neurofilaments in living neurons. At the same time, we wanted to check if this will be compatible with super resolution microscopy. So in theory, yes, it should, because we have a fluorophore, silicon rhodamine tetrazyme, which is very good when it comes to STED, super resolution microscopy. But again, we were worried if the signal to noise will be high enough to actually get super resolution imaging. And this is why you can see here first the confocal overview image and then the inset. And if you focus on the deconvolved, confocal, and STED image, you can also see these line profiles. And yeah, we get enough signal, and we also can resolve more fibers with STED compared to the conventional confocal imaging. Then we were also interested in trying to explore the fact that labeling is done in living cells, and that we can actually use different fluorophores, which means that we can establish this also for pulse chase studies, in the sense that you do first click reaction, and then after a certain time, you follow up with the second reaction. And this is interesting for us, again, in the context of axonal injury, because we can apply first fluorophore before the injury, we can then induce the injury and then label second population. And we can basically follow the fate of these proteins. And here you can see STED imaging in healthy neurons, and here now in damaged neurons. What we also try to do is to get away from transfections, so from overexpression of our protein of interest, but actually to try to combine this with CRISPR-Cas genome editing. So we first had to establish it for the wild type protein. And here we introduced a flag tag at the C terminus. And you can see here that this flag positive neuron also has neurofilaments. So we get a specific incorporation of flag tag. And then when we wanted to do incorporation of the UAAs, we actually had two options, so we technically we could have put this amber codon also site specifically. But at that point, we were worried because the amber codon suppression is not 100% efficient. And we were worried if we put it site specifically, in the absence of successful amber codon suppression, we would actually have a knockdown of our protein of interest. And that's why we decided to put it actually at the end of the gene, just before this flag tag. So this means that even in the absence of the UAA, we will have a full-length NFL. And in the presence of the UAA, we will have also translation of the flag tag, which you can see here with the immunostaining. And at the same time, we could show that these neurons then also get successfully click labeled. So yeah, this was first part of the story, and we are now still trying to improve this further in the lab and also try to adapt it for even more complex proteins. So we have here a voltage-gated sodium channel, which is more than 3 times larger than neurofilament. So it's really like a monster protein, has a very complex structure, and it's very difficult to find permissive sites for labeling. But we have managed to find some. We also did electrophysiological characterization of these clickable constructs. And we also did click labeling of some epilepsy-causing mutants. And at the same time, we also developed some viral vectors trying to increase the efficiency of this whole system. And this is also something that we published as a preprint, and that we are currently trying to finish our revisions, and we hope to see it published soon. So yeah, just to summarize what I think are advantages of unnatural amino acid-based fluorescent labeling. So really, if you have a protein which is sensitive to conventional labeling methods, this is a good alternative, because you will be changing only one amino acid, introducing minimal perturbations. Also, once you have it established, it's very easy to switch between different fluorophores, even in the same experiment. So, depending on the microscopy technique that you want to use, you can easily pick the best fluorophore for STED or STORM or whatever your microscopy technique you want to use. Also, as I already told you, this is site-specific, so it makes it very likely that for whatever protein you are interested in, you will be able to find one permissive site. And also, this type of labeling is very versatile. And what I mean by that is that, yeah, I was mainly talking about clicking fluorescent dyes, but of course we can click many other different types of probes to these proteins once we have UAAs incorporated. And also, of course, which is very exciting and also covered in this webinar series, there are many other types of unnatural amino acids which can be used for other very exciting applications. And so I just want to briefly summarize here. So what do you need if you want to set this up? So as I already told you, you need to transfect your cells with all these components, and then you will have this genetic code expansion and incorporation of UAAs. And then in the next step, you need to apply the dye and do the click labeling. And I didn't have time now to cover this, but of course, there are also other types of unnatural amino acids. So it's also possible to incorporate UAAs which contain tetrazyme groups. But from the microscopy point of view, what we still prefer more is this approach, where we have our replicable UAA with strained alkynes or alkenes, and then we have probes which are conjugated to tetrazymes, because actually these tetrazymes can quench fluorescence of some of these fluorophores, which means that when they are not bound, they are not fluorescent, and the fluorescence is restored only when they bind to their target protein. And so basically, yes, what do you need? You need plasmids, which encode these components. So for example, we still like to use the NES PLRS system, but it's also important to say that this NES, which we have in this construct, might not be equally efficient in all cell types. So of course, it might be important to check and compare different. Then you will need a plasmid, which is encoding for your protein of interest with this TAG mutation. You then need UAAs, which are clickable and which are commercially available. And you also need fluorophores. Now we have much more of them, which are commercially available. And also when it comes to cell permeable dyes, I think the field it's looking a bit better compared to when I started. And yeah, with this, I would like to thank, big thanks goes to my lab, so to my students and postdocs. They were very brave to join me and to work on this. And of course, big thanks also goes to my postdoc lab and Edward, because that's where I entered this very exciting field. And I'm very happy to take questions.
Thank you very much. That was great. I'll start off with a question. So you mentioned at the end that you're using dyes that only fluoresce once they've been kind of interacting with the protein. What is not mentioned often with fluorescent non-canonical amino acids is that you need to worry that when they're not incorporated, they still fluoresce, and getting them out of the cell away from, washing them out, or even when you're clicking. So how have you dealt with some of those issues so that you're only imaging what's been incorporated or clicked?
Yeah, I mean, we have the same issue also here, because yes, they are fluorogenic, but the thing is that, again, the cytoplasm will be full of free natural amino acids. And yes, so, there were some developments in the field trying to make these UAAs more hydrophilic. So yeah, this can help, but for these, we still need to evolve better synthetases, which incorporate them more efficiently. So, what we now do, we have to, depending on our protein of interest. So if you're doing extracellular labeling, this is not the problem. And that's why I would recommend it. If you can, go for that. But if your target protein is intracellular, then, we try to optimize by playing with the duration of the washing step. So for example, the night before, we try to get rid of the UAA and wash it just with the fresh medium. And we also, in our paper, try to compare, how this corresponds then to the background levels that we see. So yeah, it rather depends also basically on your protein of interest, but yeah, you try to wash them out. But if someone can make them more hydrophilic and efficiently incorporated, would be very happy to have them.
Yeah.
Because yes, these kind of stick to the side.
Yeah, it's really beautiful work. Are there any other questions from?
Hello, can you hear me?
Yep.
So actually, I want to know, is it like in terms of incorporation of non-canonical amino acids, is it impossible to have non-specific labeling? I mean to say that is always the incorporation is very specific.
So with this approach that we are using, basically the fact that we are relying on stop codons can lead to non-specific incorporation. This is one of the limitations. That's why, in the field, we are trying to develop ways to get rid of the stop codons, to try to use quadruplet codons. But for that, we need to also evolve ribosomes. So it's all kind of complicated. But indeed, I really think for a perfect system, we would need to get away from using amber codons. Because yes, indeed, the genes that normally use amber stop codon could get, you could get incorporation of UAAs instead of their normal stop. Although we also know that for the cell to stop the translation, it's not enough, it's not only the presence of the amber codon, it's also all the other nucleotides which are around. So that's why it's still more likely that at the stop codons, which are there to be stop signals, it's more likely that actually translation will stop than that you will have incorporation of the UAA. And also it has been shown that if even some read-through happens, then most of these proteins get degraded because the cell is recognizing that there's something wrong with them. But of course, this is something that also hasn't been really systematically studied in mammalian cells, but it is something that we have to keep in mind when we use this technology.
Yeah, so basically, I got some time to go through your recent paper. So I was curious, like, did you try to do some Western blot analysis of the neurons, cultured neurons, and see what kind of like do you see a non-specific labeling, or is it only the target protein? Like,
so when it comes to Western blot and neurons, the efficiency is very low. So even at the microscopy level, we don't see so much background labeling. Even when we do use these engineered release factor mutants, but of course we haven't done it really with the purpose. Okay, let's try to check how. But actually in neurons it's very difficult to even detect our protein of interest. So that's why they might not be the best system to study this. I would rather if we want to study this, try to do it in some conventional cell lines where we have also higher efficiency, and try to really see what is going wrong or going on.
I see. So I had sorry to take time. So actually, I had another question. Like in the procedure, I see that like after post labeling, like you have your post incubation with the fresh like the medium, the culture medium for like 2 hours or 3 hours. Is it specifically to minimize the background, or like what was the purpose like of incubation with the culture medium post labeling before fixation?
Yeah, so this will really depend again on your protein of interest. So for example, when we do labeling of extracellular proteins, we don't introduce this step. And in most cases, when we do intracellular labeling, yes, we do it hoping, to get to wash out some of the free fluorophores, which are also binding to the free unnatural amino acid. But it's not the must. So I think the best is, if you have your protein of interest and you're trying to establish it, the best is really to try and test, because in most cases you can immediately fix them or take them live to the microscope.
Oh, I see. Yeah. So thank you.